So what I'm about to show you are 16 diagrams that you should have had memorized for the AP Bio exam. Ideally, what you should do is I'm going to briefly explain what these diagrams are and the mechanisms behind them, and then after you watch this video, you should be getting a blank piece of paper and trying to actively recall what was on these diagrams, and you should be able to label everything and draw everything. So to go through these concepts very simply, the photosynthesis process, you should know that there are two major parts. The first is the light reaction, second is the Calvin cycle, and you should be memorizing what goes in and what comes out, as well as how the two reactions connect and how it gets recycled and the whole process overall. This is cellular respiration. It is the opposite process of photosynthesis. If photosynthesis was making the energy source, which was sugar, cellular respiration is breaking that sugar down, so glucose, into energy forms that we can use, which are ATP. So first is glycolysis, and then it goes, before that it was in the cytoplasm, but then it goes into the mitochondrion, and then it goes through pyruvate oxidation, citric acid cycle, and then oxidative phosphorylation. And in the process there are NADH and FADH2 being produced and being used, and like you should have the whole cycle within like your head. And then the next concept is prokaryotic cells versus eukaryotic cells. The main difference is that eukaryotic cells have a nucleus and that prokaryotic cells don't. Prokaryotic cells have circular DNA, when eukaryotic cells have linear DNA, and eukaryotic cells have a bunch of other organisms like lysosome or the ER or mitochondria, Golgi apparatus, and you should know what these organisms do as well. Next is ATP synthase. So if we go back to the cellular respiration part, in oxidative phosphorylation, a lot of ATP is produced through ATP synthase. It's basically having a proton gradient and then the protons go through a enzyme called an ATP synthase, and through that, by going through it, ADP and P is synthesized into ATP, and this is basically how ATP is generated. Next is the fluid mosaic model, and it is basically the phospholipid bilayer, where it has the hydrophilic heads and the hydrophobic tails and all the stuff that are embedded within it. So all these like glycoproteins, glycolipids, cholesterol, carbohydrate, and stuff. I don't think you have to memorize these individually, but what you have to know is that if there is a fluid mosaic model, the fluid part means that these embedded proteins or these carbohydrates are like free to flow around. And then mosaic means that there are like various stuff embedded into these phospholipid bilayer that creates a mosaic-like shape. Then is the mechanism of how proteins are generated within a cell. So this entire thing is basically like a part of the cell here. So what happens is, from the nucleus, the DNA is transferred into mRNA, and then these rough ER do their work and the ribosomes do their work, and then it goes to the Golgi apparatus and it does its work, and then it is whatever the product is, which is proteins that are being made, are transferred via vesicles, and then it either gets excreted out of the cell or gets used within wherever it is needed within the cell. Key vocabulary that should be mentioned here, if this were to be like an FRQ of some sort, is exocytosis and endocytosis. So exocytosis is basically, because it's exo, you're excreting it, and endocytosis is when the vesicle is swallowing the material that it's going to transport. And the next is the process of DNA replication. So basically what you're doing is you have multiple enzymes working, and you have a DNA strand which you open with these enzymes, and then you basically create new RNAs using these enzymes. And you should have a basic idea of all the names like DNA polymerase, helicase, topoisomerase, and etc. But and the lagging strand and the leading strand. And just like in general, you should have an idea of how the process goes. So basically the DNA is split open, think of it as like a zipper, by these two enzymes, and then there's a lagging strand and a leading strand. And for the leading strand, the DNA, like the RNA, is synthesized in like just one direction like this. But for the lagging strand, the RNA is synthesized in little segments called Okazaki fragments because of the direction it is in. So keep the whole process in mind. And the next is basically this process, but we're putting names on it, like transcription and translation. So first of all, when you open up the DNA and make the RNA, that is called transcription because you are pairing these base pairs up. And then with the RNA that you have created from transcription, you translate that into amino acids, which create the polypeptide. So every 3 base pairs are called codons, which make up one amino acid, and this sequence determines the polypeptide. So you should have memorized the vocabulary, transcription, and translation. And then next is mitosis and meiosis. So think of mitosis as creating replicas, and meiosis as creating gametes, which are known as sex cells. So remember, mitosis, it's just going to go through the process of interphase, prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis just once. But for meiosis, it's going to go through the first steps that include all of these steps, and then another second step, because it has to separate into sex cells. What happens basically in these phases is, first of all, interphase, it gets prepared, so it's prepping time. And then prophase, so if you see, before, these are very they're not organized, but in prophase, they get organized into chromosomes like these. And then in prometaphase, they start to get ready, and the nuclear envelope here, if you see, is getting it's disappearing so that the splitting can actually happen. And then in metaphase, they line up like this, and then in anaphase, they are actually being pulled apart, and then telophase is when they are actually separating and new nuclear envelopes are being formed. And the final phase is cytokinesis, where it physically breaks apart into 2 different cells. And then for mitosis, this is the end. But for meiosis, after cytokinesis, it happens once again because it has to become gametes. And the next is the Hardy-Weinberg analysis. It's basically showing you the frequency of how many big Y's or big Y small y or small y's exist within a population, and it is expressed by a percentage. So you are basically using this formula to calculate the frequencies of the genotypes that you are given. And the next is selection graphs. So say if you have an original selection, you have to have memorized these 3 vocabularies. So directional selection, diversifying selection, and stabilizing selection. So directional selection is when the original moves towards a specific trait. So for example, if there are multiple fur colors, evolution is favoring darker fur colors, so the lighter ones are dying out and the darker ones are prospering. diversifying means that it's either becoming very light or very dark. So for example, if there are two environments, like if there is like a sandy environment, and if there is like a volcanic environment where the ground is dark or white, then the species are going to diversify in order to fit these two specific and distinct environments. And stabilizing is everything becoming like average. And all these happen due to external factors and by natural selection. The next is the pyramid of energy. So you should have these terms like decomposers or producers, primary producers, secondary, tertiary, or apex predators have memorized and have a general idea of how this pyramid is formed. And you can read these examples to get like an idea of which animals specifically fit into, not animals, but organisms fit into these categories. And here it says first trophic level, second trophic level, and third trophic and fourth trophic level. The trophic here comes, think of it, associated to autotrophic and heterotrophic. So it basically means autotrophic are like plants. They make their own food by themselves, and heterotrophic are the ones that eat other animals or eat other organisms to get their energy. So trophic means like food or like where they get their energy from. So first trophic is basically like they're gonna produce it themselves. Second is they're gonna get it from somewhere else. So that helps me understand it, so it probably will help you understand it as well. And next are the biogeochemical cycles. So there's the carbon cycle and the nitrogen cycle. These two cycles are what you should have like understood. It's basically how the stuff that gets produced like is a big cycle within the environment. Carbon obviously is produced by plants, animals, factories, like human activity, and then it goes back into the ground to create fossil fuels or dead organisms, and then it comes back out and it's like a cycle. In case of the nitrogen cycle, so plants and organisms need nitrogen to sustain their lives. And the nitrogen form as an N2 is very hard to use, so it gets fixated and it turns into different forms in order for the plants or like bacteria to be able to use. And then the last one is the lac operon. So lac operon basically is, if you were to think of like if you had food and you have a person and you have a kitchen. So what the lac operon is basically is when there is food, the kitchen's light is going to turn on so the person can eat the food, right? If there is food within like a system, then the lac operon is what turns on the light so that the organism or like the cell can eat the food. So how it basically works is there is a cap and a cap site. So the cap binds to the cap site and then the promoter works, and then when the operator, when it doesn't have a repressor, allows the lac, Z, Y, and A, these are the things that actually eat the food, to work. And that is basically how it works. So after you watch this, make sure to get like a blank sheet of paper and try to kind of like draw all these diagrams out from scratch and try to like fill in the blanks for what you don't know completely, because these are like very essential diagrams and knowing these will help you a lot in the test.